and validation, and solubility enhancement techniques. She has guided over 40 MPharm students and has an impressive scholarly record with 75 research publications in national and international peer-reviewed journals. She has also authored 15 book chapters and has been honored with several awards, including the Dr. P.D. Patil National Award for the Best Thesis in Pharmaceutical Sciences to 2024, reflecting her excellence in research and mentorship. Her students have consistently excelled at prestigious platforms, receiving bronze medals and consolidation prizes at the 18th and 19th Avishkar Maharashtra State Inter-University Research Conventions and securing a consolidation rank at Avishkar. They have also secured the third prize at the Student Research Congress 2023-2024. Dr. Suvarna has successfully received industry grants amounting to rupees 8 lakh, further highlighting her commitment to advancing the pharmaceutical research. She also is a 2% scientist of the world in Stanford University list. We are honored to have Dr. Vasanthi Suvarna with us today.
Thank you.
Thank you, Dr. Vasanthi.
Thank you, ma'am.
I must also mention to all our judges that we have parallel sessions going on for AI in healthcare and hackathon on solid orals, innovation in solid orals. These two parallel sessions are ongoing along with the PhD presentation. Over to you, Ting.
Before we begin, I request all participants to carefully go through the instructions displayed on the slide. The presenters have to rename themselves as per the allotted codes if not already done. The presenters are requested to stay online, switch on their camera when the code is called out. Pre-recorded video will be presented on the screen. Participants cannot reveal their names or any affiliations. Each participant is requested to stay alert for their roll call. All the participants should be present for their session. All members from the group are allowed to answer during the Q&A session. The duration for the Q&A session is limited to 5 minutes.
Now, with the permission of our esteemed judges, we are ready to begin the oral presentation competition. Our participants and audience are eager to experience the innovative and impactful ideas presented today. Also, I kindly request the judges to confirm that they have received the scorecard.
Yeah, we have received. I have received.
Yeah, I have received.
Thank you very much. Now, I would like to invite code number PhD 006. Please be ready. We'll be playing your video shortly. PhD 006, please switch on your camera. Hello.
My identifier number is PhD 006. In many parts of the world, access to fast, accurate, and affordable diagnostic tools remains a challenge. Disease linked to liver and bone disorders often go undetected due to the lack of early diagnosis. What if a simple piece of paper could change that? Hence, we came up with a prototype titled Affordable Paper-Based Fluorescence Sensor for Alkaline Phosphatase Detection, a game changer in point-of-care diagnostics. Current diagnostic methods for alkaline phosphatase relies on expensive instrument, lab-bound system that require skilled persons. They are time-consuming, costly, and unsuitable for field or rural use. We present a low-cost, paper-based ratiometric fluorescence strip that detects alkaline phosphatase with high accuracy. The entire kit built using a commercial available chemicals and materials. This innovation costs less than 10 rupees per test and require no instrument or synthesis. The sensor works on principle aggregation induced emission. It means dye alone shows negligible fluorescence in solution because of non-radiative processes, whereas it shows strong fluorescence, especially upon aggregation, due to the block in torsional relaxation. High sensitivity, large stroke shift, and excellent photostabilities are an advantages of AI dye over conventional fluorescent probes. We use AI dye tetraanionic TP4C, cationic polymer PAA, and tri-negatively charged ATP. Our target enzyme is alkaline phosphatase. And a tetranegatively charged dye TP4C alone shows negligible fluorescence, but forms strong fluorescence aggregate with cationic polymer PAA. And it is because of the block in torsional relaxation. In this solution, upon addition of ATP break the aggregate and resulting into quench the fluorescent signal. Here, ATP is playing dual role. It is acting as a substrate for an enzyme alkaline phosphatase, and it is tri-negatively charged. Because of the electrostatic interaction, it is responsible for quenching signal off. Now, in the presence of alkaline phosphatase, ATP is broken down, restoring the aggregate and strong fluorescent signal. This reversible process forms the basis of ratiometric sensing, where fluorescent signal turn on as well as turn off. Because of this, it is self-reliable, self-calibrated response. The mechanism of paper-based assay relies on simple yet powerful drop casting method we have developed. We use reagent sequentially, dye, polymer, ATP, and different concentration of ALP. Dye alone is negligible fluorescence on the paper, whereas dye and polymer gives highest fluorescence. Upon addition of ATP on dye polymer complex, fluorescence signal quench. This quench signal will be an reference point for alkaline phosphatase detection. In the presence of alkaline phosphatase, ATP is broken down, reforming the aggregate and causing strong fluorescence signal. We use lower concentration to higher concentration of alkaline phosphatase. We optimize and validated this strip in buffer and 5% serum. Then we translate the entire mechanism into portable lateral flow assay kit, where one spot acting as a control and another is acting as a sample. On the sample, we will load unknown concentration of alkaline phosphatase, which is present in human serum. With this, we will take photo in smartphone camera, and this photo we will analyze in digital color analyzer softwares. Through software, we will find out the concentration of alkaline phosphatase using calibration plot. This ratiometric change allows real-time and highly sensitive detection. Key benefits and features are all materials are commercially available, equipment free and simple fabrication, smaller volume of solution is required. The kit is ratiometric and calibrated output. No ink or printing or paper modification is required. Hence, it is cost effective, sustainable and scalable. Here we can see that under UV luminance, we will take the photo of the strip. With this photo, we will analyze this in digital color analyzer through mobile and find out the concentration of alkaline phosphatase present in human serum through calibration curve. Now, scopes for commercialization. Filter paper-based platform can be used in environmental, food, and disease diagnostic by adapting to different enzyme targets. Integration with smartphone compatible module enable real-time and user-friendly diagnostics. With the standardized quality control procedure, the system can be reliably scaled for bulk manufacturing. Clinical validation through extensive trials with the diverse patient sample will help to establish its accuracy and reliability. Regulatory approval for securing essential certifications, including ISO and FDA clearance, to ensure safety, performance, and readiness for the market deployment. This work is appreciated in Gandhian Young Technology Innovation Award 2024. Thank you so much for your patience and time.
I now invite our judges to ask their questions to the presenter.
So how it is superior than the existing detection system? How currently it is done, alkaline phosphatase evaluated?
Sir, sorry, sir.
I am asking, currently also we detect the alkaline phosphatase, right?
Yes.
In the sample. So how this is more sensitive, specific, and accurate than the existing one? And what benefits are
Okay,
there
sir.
compared to the existing technology? Are you getting my question?
So in institutes, either spectrophotometric based. Yes, sir. Am I audible?
Your voice is breaking in between.
Am I audible, sir?
Your voice is breaking.
Now, if oh shit.
Speak a little loudly.
Now,
Yes.
yeah. So current existing technologies, either SERC, that surface enhanced Raman spectroscopy, or ELISA, or colorimetric methods are used. In colorimetric method, they are it is using with PNPP, that is for the PNPP is another substrate of alkaline phosphatase, but in the in presence of high temperature or either high temperature or any other environmental parameters which suppress this sensitivity in case of colorimetric sensor, whereas in surface enhanced Raman spectroscopy, it is a costly instrument and it required labor, which is trained labor. So in this filter paper based method, we are just drop casting this dye and polymer. With this, we are using ATP, which is known substrate of the alkaline phosphatase. So alkaline phosphatase will break, and as I said, the substrate is ATP. So it is more specific, and there is no environmental parameters are involving for this detection. Like I am saying, environmental parameters means that the temperature or any other parameters are not matter in this case. That we have tested this sample with different pH, different temperature, and then we optimize this.
Okay. Thank you. Over to you, Dandas. You can take.
Dhanraj, sir. Sir, you're muted, I think.
It's not muted. We are not able to hear.
Okay. Not able to.
Sir, we are not able to hear you, Dandas. Okay, till he joins, one question to you, one more. Usually paper-based tests are preferred for non-invasive methods, where the sample is you don't have to invade a patient. So here it is like a plasma sample. So how it will you think that anyways for plasma we are taking it for ELISA also. So why one should go with the paper based?
Sir, in ELISA, it is time consuming as well as it is trained labor is required.
But
That is
anyways,
main
for liver
disadvantage
function,
there.
time is not a constraint. Anyways, the patient
Yes,
can
sir.
wait. It doesn't take much time to get liver function to be analyzed.
Yes.
So I'm
Sir,
just
other
looking
than
from a clinical relevance, whether what you are doing has
okay, sir.
translational value, right?
Sir, another in my future direction, I am going to test this sample with the saliva whether we can go saliva because we are thinking about invasive methods. So saliva can be another source of alkaline phosphatase. It is I am proposing this so that it will be eliminate invasive like taking serum or blood.
Okay. So I think time is
Okay, I have
over for that.
just one question.
Yes, sir.
Like, what do you think is the precision of your experiment? Precision means like the detection capacity.
Sir,
Okay.
we are using fluorescent dye. We are using fluorescent dye. So this dye, the very less volume of dye is required, and in that we are using in UV. So there, this fluorescent dye is getting in the presence of UV light. We can observe the color develop.
Okay.
This is
What
the
about
main
what?
mechanism. And now I'm going to. Sorry, sir.
What about the half-life of the dye? Does it, decolorize fast, or like is it it retains for a long time?
Yes, it is retained for last time. So we have did this 8 months ago, and we compared the 8 month the initial strip with the 8 month before strip, and this I have data that the dye color is not much changes. So the fluorescent complex developed on the paper it remain same as it is in initial.
Okay. Thank you.
Yeah,
Thank you, sir.
one thing from my side is the publications which are there on this. You said no PNPP is used, which is not a natural substrate. Now you have used natural substrate, yes, it has increased the reaction chances. But what about the ATP, ADP, which is already present in the serum matrix, the sample which you take? How will you make sure that it's not interfering?
So, ma'am, ADP and inorganic phosphate have less charges than ATP. And my dye, which is tetraphenylethylene, it is a 4 negatively charged. So this, because of the 4 negatively charged, there are chances that this dye will competitively bind with this polymer and not ADP and inorganic phosphate because of the less charges. Since this entire mechanism of electrostatic interaction, so higher charges will dominate the system.
So you don't require ATP for the activation?
No, we are just the main what we can say. It is the fluorescent platform. It is depend on the dye and polymer, not on ATP. ATP is just a substrate, which is we are going to add with the.
Sorry for interrupting, but we would like to conclude with the presentation now. We would start with a new one.
Okay, thank you.
Now I would like to invite code number PhD 009. Please be ready. We'll be playing your video short. Please switch on your camera. Please switch on your camera.
Cardiovascular diseases and which often lead to increased morbidity in extraction presentation of presence and absence of jasmine humule. So I have tested numerous phytoconstituents, starting with the alkaloids to the fat and fixed oils. And this is a representation of presence and absence of the phytoconstituents. So minus sign represents the absence of the particular alkaloid in that particular phytoconstituent in a particular extract, and plus sign shows the presence of the particular phytoconstituent in the extract. Double plus sign shows the moderate presence of the phytoconstituent in the extract, and triple plus sign shows the maximum quantity or the maximum presence of the phytoconstituent in a particular extract. Next, for the clarification, I have done the thin layer chromatography. So these are the photos of the TLCs. First is the ethyl acetate, then the PET picture, then the chloroform. At last is the ethanol. For the TLC, I have chosen the solvent system of acetone, ethyl acetate, and formic acid within the ratio of 5:4:2. The next is the DPPH radical squeezing activity. So DPPH is basically done for testing the anti-obesity activity or antioxidant activity of the particular compound. For this, I have taken the standard of ascorbic acid, and the maximum phytoconstant present in the ethyl acetate and the ethanol extract. So I have taken these two extracts for the further study. So the blue line shows the DPPH activity of the ascorbic acid. The red line is of the ethyl acetate, and the last COH, which shows the presence of a phenolic or a polyphenol presence in the ethyl acetate extract. Next, I have done the screening, or I have prepared the nano emulsion. So for my in vivo study, I have taken 5 groups. Number one is normal control group, in which no drug has been. Number one is normal control group, in which no drug has been given. Then high fat diet control group, in which I have given only the high fat diet without any drug treatment. Then is high fat diet plus orlistat. Orlistat, I have taken the orlistat as a standard drug. After that, the high fat diet plus J humule extract low, that means the low dose of the J humule extract nanoemulsion. After that, last one group is high fat diet plus J humule extract high, that means the high dose of the jasmine humule. So initial weight of the normal group is 260, then is final weight after the completion of the 6th week is 264. That means there is a no significant variation in the body weight in the normal control group. After that, high fat diet control group, the starting weight is 237, and by the end of 6th week, the weight increased to 297. And then is high fat diet plus orlistat, that is 244, and after that 285, and so on, as present in this table. After that, last I have done the histopathology of the right kidney and right kidney of my animal. So first of all, I have done the histopathological study of second group, that is control group, then is the high fat diet plus orlistat, then the third group, jasmine humule, low dose, and at last jasmine humule, high dose. Next is the conclusion. So the low value of PDI demonstrate the formulation are in monodisperse in nature. The zeta potential of nanoemulsion formulation was found to be minus 24.4 megawatt, which signifies the stability of the nanoemulsion formulation. Formulation showed the percent drug release of 96%. The pH was between 5.43 to 5.67. The developed J-humule nanoemulsion showed the viscosity in the range of 22.1 and 22.3. The best fitted kinetic model is Cosmiar-Papas model with the regression coefficient value of R scale 0.9502. The pancreatic lipase activity was performed. Orlistat gave an IC50 value at 25 microgram per ml concentration, and jasmine humule ethyl acetate extract gave an IC50 value at 100 microgram per ml concentration. Stability study was performed for 3 months and the result showed the formulation is stable. In vivo, studies were performed to check the anti-obesity activity of J-humule extract nanoemulsion compared with the standard orlistat. The J-humule nanoemulsion at low dose that is 100 mg per kg, demonstrated significant anti-obesity activity in high-fat diet-induced obese female. This finding confirmed that jasmine humule leaves extract has potential as a natural anti-obesity agent, particularly when delivered through nanoemulsion technology. Nanoemulsion enhances the bioavailability and efficacy, leading to improved anti-obesity activity. This research contributes valuable insights into development of novel plant-based therapeutic strategy for managing obesity. Next, these are the references. And thank you very much.
Participant number 009, kindly switch on your camera. I now invite our judges to ask their questions to the presenter. Dear judges, since participant number 009 has not switched on their camera, we would now like to proceed.
Whether she's there or not.
No,
We are
she is
going
not
to the
there.
next.
She has not joined the meeting.
So we have to go to the next presentation, right?
Yes.
Now, I would like to invite code number 010. Please be ready. We'll be playing your video shortly.
Hello everyone. Today I am here to present my oral presentation about formulation and characterization of burgeoning nanoparticles and its evaluation on adjuvant-induced arthritis. So start with introduction. Introduction is about rheumatoid arthritis. Rheumatoid arthritis is defined as the systematic autoimmune pathology associated with a chronic inflammatory process which can damage both joints and extra-articular organs, including the heart, kidney, lung, digestive system, eye, skin, and nervous system. Arthritis is classified into non-inflammatory arthritis like osteoarthritis and inflammatory arthritis caused by crystal deposition like pseudogout, basic calcium phosphate disease, gout, by bacterial and viral infections like Staphylococcus aureus and Neisseria gonorrhea, complication of Lyme disease, parvovirus, and enterovirus, or by autoimmune system. You can see here the normal hand and the rheumatoid arthritis hand. They are different with each other, and depositions of crystals in the joints. And the next is rationale of study. Virgin comes under biopharmaceutical classification system of class 4, hence its poor solubility and low bioavailability. Virginin is a class 4 drug and has proved its poor solubility and viability, and to increase its viability, making the formulation of nanoparticles and increase the solubility and also viability because they are smaller in structure and have a big systemic circulation or high surface area and increase the therapeutic efficacy also. Due to low solubility and dissolutions dependent viability of virgin, it affects the therapeutic efficacy of the drug. Virgin undergoes significant hepatic first-pass metabolism, resulting in decreased amount of the compound reaching the systemic circulation. This phenomena ultimately leads to the reduced oral bioavailability in normal rats, consequently impacting the efficacy of the substance. It increased the effectiveness of poorly soluble drugs. Researchers have been focusing more on new solubilization technology, such as synthetic water-soluble products, nanoparticles, and phospholipid complexes. The development of suitable oral drug delivery system in a research focus of virgin nanoparticles. Nanoparticles can significantly increase the solubility and permeability of the virgin and improving its solubility and bioavailability. This means that a smaller dose of virgin can produce the desired therapeutic effect and reducing the risk of side effect. Administration of high dose increasing the cost of therapy and poor patient compliance. By reducing particle size, virgin nanoparticles, the dependent bioavailability is enhanced. Anti-arthritic activity of virgin-loaded PLGA, polylactic glycolic acid nanoparticles are not reported till date. Thus, this project has taken into consideration. Here is the methodology. We start with pre-formulation studies of drug, optimization, formulation of virgin nanoparticle, characterization of virgin nanoparticle, in vitro drug release, stability study, and in vivo study. In pre-formulation study of drug, we carry out organoleptic properties, melting point, calibration plot, pH determination, partition coefficient, dissociation constant, determination of solubility, FTI spectra, and PXRD. In optimization, we used the design expert of Statis company, and the design software is 12.0.7 version, and the formulation of virgin in nanoparticles. We the solvent evaporation method. We use solvent evaporation method to produce or formulate the nanoparticles, and then characterization of virgin part nanoparticle. We check the nanoparticles, particle size, zeta potential, bodily dispersity, index, and drug content, entrainment efficiency, drug loading, scanning, electron microscopy, and transmission electron microscopy. FTIR to check the functional group, and XRT to check the crystalline and uniformous nature of the drug and nanoparticles or PLG loaded, and then the drug release of nanoparticle by using the method of dialysis membrane. The next is stability study. How was the stable the drug is, and the last is evaluation study, in vivo study. Here is the results of our virgin nanoparticles, the particle size and zeta potential, FTI spectra analysis, HRTM, and FESAM in vivo evaluation, the radiographic analysis of the animals, and the spleen index assay. And these are the induction of the CFA of arthritis inducing the rat in bow, and the histopathological examination of the in vivo studies. And next is the result. The virgin loaded PLG nanoparticles were prepared. Various pre-formulation studies has been done. Organoleptic properties, melting point, calibration plot, pH determination, partition coefficient, dissociation constant, solubility studies, and FTI spectra, PXRD, and characterization of the virgin loaded nanoparticles are zeta potential, particle size, body dispersity index, drug content, entrainment efficiency, and SAMDAM. In vitro drug release was found to be 91% within 72 hours, and enhances solubility, stability, and viability. And also observed is therapeutic effect. And this could be particularly useful for delivering poor solubility drug to improve the therapeutic efficacy of virgin itself. Nanoparticle can be designed to target specific tissue or cell in the body, allowing for target therapy. And then is in vivo studies were performed to be investigate the improved anti-arthritic activity of virgin loaded nanoparticles compared with standard endomethacin and virgin group. Virginal loaded PLG nanoparticle demonstrated significant anti-arthritic effect, no observed toxicity in animal model. This finding indicates the virginal loaded PLG nanoparticles could be valuable options for further arthritis therapies. And these are the references. And thank you very much.
Judges, we would like to tell you that participant 010 is not available in the meeting. So we would like to proceed with the next session.
Uh, Here I have one suggestion. If the candidate is not present, that means he is absent. We should not even run his presentation.
Okay, so thank you for your
Because it is
insights.
just wasting our time unnecessarily. Because
It is.
they are not serious
Yes, sir.
unnecessarily. First you check whether they are there, then only run. Otherwise, we'll mark absent in scoring. We are not giving any marks to them. At least I'm not going.
All of the other participants are there in the meeting, so we would like to proceed with the next session.
Now, I would like to invite code number PhD 011. Please be ready. We will be playing your video shortly. Please switch on your camera.
Hi everyone, I'm PhD 011, and over the course of this presentation, I will be focusing on transcending experimental constraints using in silico characterization of viscosity for self-assembling nanoparticulate systems, where we'll be studying these systems at both nanoseconds and microsecond timescales. So first, I'll just walk you through the presentation where we'll just go over the background and rationale, the methods on how we did the experiments. We'll discuss the results, data analysis, and lastly, end with a conclusion and outlook of the entire project. So we have used SLNs, also known as solid lipid nanoparticles, as our model nanoparticulate system. Over the recent years, over the last 20, 30 years, we have had different nanoparticulate systems, such as liposomes, SLNs, nanostructured lipid carriers, and many others, which have been emerging as promising drug delivery platforms. However, in this, we'll just be talking about SLNs, which were initially introduced in the 1990s. And over the past 3 decades, they have redefined drug delivery, moving to multifaceted drug targeting of the nanoparticulate systems, their biocompatibility, biodegradability, stability, both in vitro and in vivo, as well as controlled release properties. So these are usually submicron particles and often fall in the range of 50 to 500 nanometers. And they often are used for protecting and targeting usually BCS class 2 drugs. However, recently BCS class 3 and 4 have also been explored. While a lot of people know about SLNs, the most important facet that we will be considering here is the viscosity measurement. Because when we are looking about for commercial manufacturing, especially when it's of the magnitude of 200 mils per minute, using microfluidic systems or high shear mixing and stuff like that, it is really difficult to maintain the viscosity. And since it is continuously self-assembling, we need to keep a close eye on the viscosity of SLNs and other nanoparticulate systems. So why do we do this study? So the dynamic viscosity of nanoparticulate systems is really important because these systems continuously self-assemble and rearrange, often altering the microstructure and also changing the flow resistance of the fluid injected over time. Oftentimes these changes are of the nanosecond to microsecond scales, and often in the magnitude come similar to molecular collisions and Brownian motion, which often makes them highly transient. And we do not at this moment we really do not have the instruments that can measure these changes both on a temporal, that is a time, and spatial, that is the nano resolution. although the viscometers have been developed, but they cannot really measure on an instantaneous scale. They usually take certain time, and that is why we are using in silico characterization. So in silico, whenever we think of in silico, the first thing that people would think is using an all atomistic study where you take the entire molecule and it is in its atomic state, and then you go for simulations. However, that is not very practical when it comes to you have 100 million molecules or 100,000 molecules. Um, so in that magnitude, when you're trying to study the system and the bulk system, it is really very complicated for all atomistic simulations to take place, and it is not computationally viable. So in such situations like that, we use certain thing called as a coarse-grained molecular dynamics, also known as CGMD. So we would use this for analyzing the behavior and property trend for in silico thermodynamic self-assembly of SLNs. So why use CGMD? CGMD is efficient when it comes to studying the self-assembly of really large molecules like lipids, bigger drugs, proteins, and often it is beyond the reach of atomistic models. And second is it faithfully represents the thermodynamic and structural behaviors of most of the components inside the system, and often inform us how the formulation choices would impact the size and stability of SLNs. Here we'd be studying viscosity. So how do we identify the parameters? First would be something what we call as mapping. We take the entire molecule, and these molecules include things like lipids, which in our case is glycerol diphenate, surfactant, which is pluronic F127. Then we have a drug that is ibuprofen, and we have water and antifreeze. So the entire antifreeze is required because we often start the simulation from 0 Kelvin, because that is where the entropy is 0. So when it comes to studying the systems, it is really difficult to bring the system back to 298 Kelvin, that is roughly around 73 degrees Celsius. So that is why we use antifreeze. And we would be mapping the entire molecule for 4 atoms, that is 4 heavy atoms per CG bead, which means that every atom except that of hydrogen would be treated as a heavy atom. Like say 4 carbon atoms would be treated as 1 bead, or 4 water molecules, that is 4 oxygen molecules, would be treated as 1 molecule of water, 1 CG bead of water. So this would give us a balance between molecular detail and the computational tractability. Second thing that we'd be looking for is force field parameterization. Herein, we would use something which is called as a bottom-up approach, where we will form an atomistic simulation of 1.5 microseconds. And we would be using a highly parameterized force field called as the OPLS4, that is Optimized Potential for Liquid Simulations 4. And from here, we would be extracting all the bonded parameters, such as bond lengths, angles, dihedrals, improper dihedrals, and non-bounded interactions such as van der Waals forces, electrostatic forces, that is the Lennard-Jones potential. And all these distributions would be taken from the atomistic study. And like we have machine learning, we have something called as iterative Boltzmann inversion, IBI, where we refine the force field parameters over 90 cycles. For example, if you can see in the study in figure 4, you would be able to see that the red line is the reference analysis that we get from atomistic simulations. And the bigger peak, the light gray peak, is the one that we get from the first fitting cycle. But overall, when we are trying to go for 90 fitting cycles, that is where you find the black line. The line would show that there is not significant difference between what you see in a CG simulation, that is a coarse-grained simulation, versus an atomistic simulation. Subsequently, we would be taking parameters from the best practices for MD simulations, where non-burdened cutoff parameters are fixed. We keep the electrostatics, van der Waals, and hydrogen bondings at 12 angstrom, 7 angstrom, and 4 angstrom cutoff. Subsequently, we'll be converting the potential from Lennard-Jones to shifted Lennard-Jones potential, as you can see in figure 5. That you would see that the green line indicates that instead of the blue line, the green line is indicating that the potential reaches 0 after a point of time, which is not the case with the standard Lennard-Jones 612 potential. Subsequently, we'll be running the simulation for 1500 nanoseconds. That would be 1.5 microseconds to see the self-assembly. Lastly, after the system would be equilibrated, we're using NVT or NPT, where the number of molecules, the volume, temperature, or the pressure and temperature are stabilized. We would be running two different systems. One would be measuring using the Green-Kubo integral, which has been the gold standard for a lot of systems. Herein, we'd be running an equilibration MD simulation, and we'll track how the internal stresses fluctuate and relax over the time. So you put stress on a particular system and you allow it to relax. The system would come back to normal and you would get some forces. And these fluctuations, if you keep on doing that for a couple of times, you would get a lot of fluctuations. And that gives you the internal resistance or the viscosity of the liquid. Second, we'll compare a more defined model called as the Einstein-Helfand method. It is predominantly not frequently used for simulations. Herein, from the same Green-Kubo integration simulation, we will build something called as a stress displacement metric, also known as a RMSD, and see how it fluctuates over time. The slope of that would help us get the viscosity. Lastly, we'll be correlating the data with experimental data from the viscometer. And lastly, statistical analysis would be run in place. So moving on to the results, you would see that on the left, you would be able to see glyceryl dibehenate, which is an all-atomistic representation. We simplify that entire complex molecule, with a lot of carbon atoms and stuff. We try to put it down to a simple coarse-grained representation. Second is you would see ibuprofen on the left and ibuprofen on the right. So on the right, you would be able to see a more simplified version of each of these molecules. Next, we have made a representation of Pleuronix F127, which is not the exact molecular weight, but is in the same ratio of the actual Pleuronix F127. Lastly, we take in water and convert that into the coarse-grained representation, that is what you see in the green small bead. Also, antifreeze water is there, and antifreeze water is nothing but water, which is non-polarizable. So that doesn't allow the formation of hydrogen bonds. Next, we put all of these systems in an entire box where we have over 120,000 molecules, of which it's roughly around 400 molecules. That is 10% by weight of lipid, 1% by weight of drug, and 4% by weight of the surfactant, as we use for general synthesis purposes or nanoparticulate system assembly. We try to put it, we assign the force field parameters that we have developed with the iterative Boltzmann inversion. You can see when we run the simulation, it would start at 0 nanoseconds. It's a really unbiased scattered system. When it is 300 nanoseconds, you would see that there are two different smaller nanoparticles, SLNs, that have been formed. But with the end of 1500 nanoseconds, it coalesces to form one single blob. And I think that should be one single SLN because it is more like a mirror image. So the smaller part that you see on the right would eventually shift to the left because it's just a snapshot of an actual moving movie or a film. This viscosity analysis has been summarized here, where we use Green-Kubo integration method and Einstein-Helfand integration method. We measure it in 3 replicates for a duration of 80 nanoseconds after equilibration and after the self-assembly process. For Green-Kubo, we are using an exponential fitting method, and we obtained a shear viscosity of 0.55621 centipoise with the standard error of 1.7 into 10 power minus 5 magnitude. Since we have done 3 replicates, it is important to see an intra-class correlation, and we found that since it is greater than 0.70, it is a strong correlation. And we wanted to test the correlation with the in vitro studies. So for that, we had to check for things like the Shapiro-Wilk normality test and the Levine's test for homoscedasticity. which both of them did pass as the null hypothesis. For the null hypothesis, it is showing that there is normality and homogeneity since the p-value is greater than 0.05. Subsequently, we did a Pearson's correlation, product moment correlation, and we found that between experimental and the in silico value, there was a correlation of 0.7899, roughly around 79% correlation. In contrast, the Einstein-Helfand method had a shear viscosity of 0.433 centipoise and a standard error of 5.6 into 10 power minus 5. Again, it had a same strong intraclass correlation and passed both the normality and homoscedasticity tests. However, the intraclass correlation using Pearson's product moment correlation was 0.8566 or 85.66% correlation, often suggesting that Einstein-Helfand method is a better way to measure the viscosity of dynamic and self-assembling coarse grain systems. And we have optimized the entire system to fit, what is much more needed. And it is an optimized system that you're looking at. So lastly, with this, I'd like to conclude that, we have developed and validated a robust protocol for the development and in silico measurement of viscosity. And we are used it to compare the effect of pressure tensors in various directions, that is X, Y, and Z axis, and also while varying the formulation parameters. We have checked for internal two-way consistency in the results using ICC and reported the reliability analysis in the previous slide. Subsequently, in the outlook, we plan to study the effect of formulation parameters on the final SLNs, especially by measuring things like the surface properties, the curvature, and eventually visualize them in silico. Well, these are the references that I have used over the course of the presentation, and they have been duly cited. And with that, thank you for being such a lovely audience.
And now invite our judges to ask their questions to the presenter.
Hi, everyone.
Not able
Hi,
to
Dr.
hear.
Dhanraj. I'm sorry, but I'm unable to hear you. I'm sorry, I can't hear anyone. Yep.
Sir, you're muted. Please unmute yourself.
What is the application of your work? Clinical value, clinical translation.
Oh, well, thank you so much for the question. The actual applicability is beyond clinical use. It's predominantly in the manufacturing sector where you had the mRNA-based vaccines, predominantly what we used for the Pfizer COVID vaccine. That's the mRNA vaccine. The Moderna vaccine, they were predominantly used using ionizable LNPs, so which again is made by the microfluidic systems. And oftentimes you would find that it's roughly of the magnitude of 2 liters to roughly around 20 liters per minute on a commercial scale. So in these cases, it is often very difficult to channelize the entire viscosity because the viscosity keeps on changing. And as you pass it through the entire channels of the microfluidic system, you would eventually see that the viscosity changes. So it has a more commercial implication. And yes, eventually, yes, it does make it to the patients. So that is why we really need to ensure that the product that we're making is pretty much safe. It's pretty much standardized. So, well, I hope that answers the question.
Yeah, there was what I was concerned about. Now I think I'm audible.
Of course you are.
Yeah. So you have done the Green-Kubo method and Einstein-Haffel this one method for viscosity. So
Yes.
I could see that when we go towards the this one standard error,
Hmm.
standard error was more than 1 point something on one side, and on the other side was more than 5.
Yes.
So you need to work more on that. You need to increase the number of experiments, or you need
Yes.
to you need to improvise the self-assembly thing, provided you take care of the viscosity also, consistency of the viscosity. Right.
Absolutely. That's a really
So
good comment. Yeah.
yeah, what I want to ask one more question is that, like, what was the thing which was interfering the most when you ran the experiment? Like, is it the this one van der Waals force or hydrogen bonding, or what is it?
Well, I would like to say that the thing that concerns me the most would be the Leonard-Jones potential, because
Okay.
we do not have many charged base moieties in the system that we're using. So it is very limited by electrostatic interactions or the Coulombic forces. But predominantly, we would see the van der Waals forces and the Leonard-Jones 612 forces that predominantly govern the self-assembly.
Okay. Thank you.
Yes, of course.
What are the limitations of your MD model?
Well, it is a very interesting question to look at, and thank you for the question. The major limitation that I can think of is, since these models are like unlike atomistic models, which are deterministic, which means you run it for 100 times, you'll get the same result. On contrary, these are called as Langevin models, Langevin dynamics, which are stochastic models, which predominantly run on probability. So like there is chance like 70-80% of the chances you'll get the same result. But there may be an unforeseen condition where you might see something that's deviated, but more or less around the same lines. So you like the reliability of it is it's a bit skewed, I would say. That is the predominant, like the major limitation that I can think of. So
Okay.
that is why we
Data
need to
access is
increase the number of replicates, as Dr. Dhanraj has rightly pointed.
correct. Correct. Okay. Thank you.
Of course.
Thank you. Now,
Thank you
I
so
would like
much.
to invite.
Thank you, judges, for your insightful questions. Now, I would like to invite code number 012. Please be ready. We'll be playing your video shortly. Please kindly switch on your cameras.
Candidate, please switch on your camera.
Thanks to all. The topic for my presentation is biotin functionalized mixed micelles for peptide delivery, enhanced oral bioavailability via intestinal transporter targeting. Diabetes is a chronic condition that occurs when body cannot produce enough insulin or cannot effectively use insulin that it produces, which leads to high level of glucose known as hyperglycemia. As per the latest International Diabetes Federation data, it says that approximately 11.1% prevalence is there in the world, which means approximately 589 million adults are living with diabetes, which is projected to reach approximately 853 million by 2050. And if we talk about India, there is a 10.5% prevalence of diabetes in India. And by 2050, it is projected to reach approximately 157 million. Insulin is a peptide hormone which is secreted by the beta cells of the pancreas. It has two chains, chain A and chain B, which are linked to each other by disulfide bond. It has a molecular weight of around 5808 Dalton, a low log P, and it has high solubility, which makes it a BCS class 3 molecule. It is degraded by the insulin degrading enzymes in the GIT, due to which it has a low oral bioavailability of less than 1%, due to which it is administered subcutaneously. And however, there are drawbacks of subcutaneous routes, which include patient compliance issues, hypoglycemic shock, peripheral hyperinsulinemia, Hence, to overcome, oral delivery can be explored. However, as we discussed, due to biochemical barrier, mucous barrier, and cellular barrier, there is peptide degradation, low absorption, and low oral bioavailability of the peptide. Hence, to overcome the challenges associated with the oral delivery of the peptide, here we are delivering it via biotinized pluronic F127 and bile salt mixed micelles. Here we are using biotin as it is a substrate for SMVT transporter, which is sodium dependent multivitamin transporter present on the intestine. Here it will be used for intestinal targeting, and further the bile salt used will enhance the permeation. When the formulation will be given orally, it will firstly protect the peptide from the GI degradation, and it will be transported to the intestine via these transporters, which will lead to enhanced permeation and enhanced oral absorption. These are the objectives. The first objective is to synthesize the conjugates and preparation of micelles. The second is the characterization of the prepared micelles. The third one is the in vitro release and ex vivo permeation studies. Fourth one is to perform the cell culture characterization. And lastly, the in vivo evaluation of the developed formulation. Coming to our first objective, which is preparation of micelles, the first step is to produce biotinized PF127, for which biotin was conjugated with pluronic F127, and further the amino acid conjugated bile salts were produced by using deoxycholic acid and glycine to form DG, and deoxycholic acid and lysine methyl ester to form DK. All these conjugates were characterized spectroscopically, and in this way we formulated two set of formulations, one with BP and DG and another with BP and DK. And using these conjugates, the micelles were produced using using thin film hydration method, and they were characterized. For characterization, the size, PDI, and entrapment efficiency of the prepared micelles were evaluated, and the particle size was well below 300 nanometer, and the PDI showed a unimodal distribution with a good entrapment efficiency. The TEM images also revealed spherical morphology of the micelles. Coming to the chemical and conformational stability, the page analysis showed that the molecular weight of the insulin in the formulation 1 and formulation 2 was similar to that of the native insulin. Further, the conformational stability was confirmed by the CD spectra, and it showed that the conformation of the insulin was maintained in the formulation as well. Moreover, the formulation was also stable in the biological fluids, including the SGF, SIF, and PBS. Coming to our third objective, the in vitro release study was performed using dialysis bag method in SGF, SIF, and PBS, and it showed that both the formulation showed a more sustained release effect. However, the free insulin was released approximately 100% in 6 hours. Further, the ex vivo permeation study was conducted in France diffusion assembly using the goat intestine. In the donor compartment, the formulation was kept, and in the receptor compartment, PBS 7.4 was there. And from the data, it was deducted that the formulation showed approximately 4-fold increase in the intestinal permeability as compared to the free insulin. In the fourth objective, the cell culture studies were performed. The cell uptake study was performed in CACO2 using the FITC labeled insulin, and it was seen that the FITC loaded mixed micelles showed a higher fluorescence as compared to the free FITC insulin, which showed that there was a higher internalization of the mixed micelle in the CACO2 cells. Further, to validate our hypothesis of utilizing biotin as SMVT transporter substrate, we conducted a SMVT inhibition assay using the inhibitor lipoic acid, and it was seen that in the absence of the inhibitor there was a higher fluorescence, whereas in the presence there was a low fluorescence. Further, the CACO2 cell permeability assay was carried out, and it was found that both the formulation they showed approximately 4 to 4.5 fold of increase in permeability as compared to that of the free insulin. Further, the CACO2 cell viability assay was performed using the MTT assay, and from the data, it was found that both the formulation were safer and non-toxic. Coming to a last objective, which is in vivo studies, the pharmacokinetic study were performed in SD rats. The insulin 5 units was injected subcutaneously and compared with the orally given mixed micelles. And it was found that the area under the curve of both the formulation was increased by approximately 1.2-fold as compared to the insulin given subcutaneously. Further, the relative bioavailability of both the formulation was found to be in range of approximately 22 to 25%. Lastly, the pharmacodynamic study was performed in the ST rats. The streptozotocin was induced intraperitoneally to develop type 1 diabetes, and the rats with blood glucose level more than 250 mg per deciliter were used for this study. The formulation was compared with the insulin given orally and injected subcutaneously, and it was found that the orally given solution did not produce any hypoglycemic effect, whereas both the formulation produced a hypoglycemic effect which was higher than that was produced using the subcutaneous solution, hence which was validating the superiority of our formulation. Concluding our studies, SMVT targeted biotinized pleronic bile salt mixed micelles were prepared and characterized. The formulation was stable in GI media and exhibited 6-month storage stability. In vitro release study showed a sustained release effect, and ex vivo permeation showed 4-fold enhanced permeation. The formulation showed enhanced cell uptake and permeability with low toxicity in CACO2 cells. The pharmacokinetic studies revealed better relative bioavailability, and formulation exhibited % cumulative hypoglycemic value higher than insulin injected subcutaneously. Thank you for your kind attention.
I now invite our judges to ask their questions to the presenter. Thank you.
Thanks to all.
Yes, good afternoon.
Good afternoon, judges.
Go ahead, sir.
So, like as far as bioavailability is concerned, your formulation has sought 4 times bioavailability, absorption rate. So you say that it is 4 times more, effective than the native insulin, right? So my concern is, have you studied regarding the toxicity of this particular thing on various, vital organs like kidney?
Good afternoon, sir. Thank you for the question. Sir, here we have not particularly studied the toxic effects. However, the components here we have used are biocompatible. Here we have used components such as bile salts and pleronic, which are safer, and already their safety is already registered. And secondly, in case of insulin, for it, if there is excess, it can show hypoglycemic effect. But however, in our study, there is no hypoglycemic effect is shown for our formulation.
Okay. Okay, thank you.
Okay,
So
thank
please
you,
go
sir.
ahead.
Okay. So if you see clinically, the insulin is available as a regular insulin and short acting, ultra short acting, long acting, and then it comes in a composition also, right? There are different
Yes, sir.
compositions which are available. So where do you see these micelles fall within those categories?
Sir, for these, we have studied the pharmacodynamic study till 24 hours, and the peak concentration of insulin we get at approximately 8 hours. So we could say that it could fall under intermediate. The solution which it can be comparable to the intermediate rather than the rapid or other solutions, and because it show a more sustained effect as compared to that of the subcutaneous as well.
Okay. Why did you choose biotin as the targeting ligand for oral delivery instead of other vitamins or like folate or vitamin B12?
Sir, like vitamin B12, these need a particular intrinsic factors for their activation. However, in case of biotin, there is no requirement of such case, and the transporter what we are targeting, like SMVT transporter, these are abundantly present in the intestine, so it can be easily targeted for that. Hence, biotin is used. It is like biocompatible as well, and there is no such toxic cases for biotin. And as I said, that as compared to other vitamins, there is no intrinsic factor or any activation further required.
Okay. ठीक. Thank you.
Okay, thank you, sir.
Yes. How do you address this challenge? Let's say after absorption, GI absorption by the micelles, the peptide degradation or your insulin degradation, how that is taken care of?
Ma'am, could you please repeat? Like you are asking about GI absorption or
Yes.
degradation.
Yes. So from your micelles, like once the insulin is released, right? So further degradation, how is it minimized?
We have studied the stability in GI media, and we have seen that there is the insulin which we characterized. We checked the CT spectra for the conformation change, and we did the electrophoresis analysis, and from that we checked that the micelles are stable, and due to the low CMC of our micelles as well, there is no much dilution, and the integrity was maintained in the GI media. And for the release, they were released mostly in the either by the if they would be released in the intestine, either by the micelle degradation and in the cells, the enterocytes, they are released there.
Okay. Did you put non-biotinated micelle as a control group in the study?
Yes, ma'am, we did.
Okay, and what was the
And here
analytical
we have
yeah.
we have checked that in our in vitro cell culture studies, as well as in our pharmacodynamic and pharmacokinetic in vivo studies, and we have checked we have received the superiority of the biotin conjugated formulation, and the difference we could see was approximately more than it was 1.5x than the non-biotin conjugated micelles.
Okay, thank you.
Now we would like to proceed to the next session. Thank you, sir. Thank you, judges, for your insightful questions.
Now, I would like to invite code number PhD 014. Please be ready. We'll be playing a video shot. Kindly switch on your camera.
Presenter, please switch on your camera.
Hello, my camera is on. I'm there in the meeting.
Okay.
Thank you. A warm welcome to one and all. Today I'm here to present my research entitled Exploring the Potential of an Ionic Liquid-Based Drug Delivery System for Oral Viability Enhancement of Niclosamide. We are quickly going to see exactly why, how, and what. So why niclosamide? Niclosamide is an FDA-approved antifungal agent. Recently, this drug has been repurposed for its anti-cancer potential. Despite of having a broad spectrum of activity, its clinical usage has been constrained, mainly due to its solubility and viability constraint. This drug belongs to BCS class 2, has a very poor solubility and oral viability of less than 10%. Resultant, more than 1500 mg of the drug has to be administered, which is again results into dose-dependent toxicity and compromises the patient compliance. So to tackle all this limitation, there is an urgent need of an effective formulation approach. In proposed study, we propose an idea of ionic liquid-based formulation. So what are ionic liquids? Ionic liquids are molten salts which are composed of a pair of cation and anion which remain liquid at room temperature, and they have an exceptional solubility enhancement power. They are known to alter the membrane integrity and membrane fluidity, thereby improving the permeation as well. Together, they serve as a green alternative or green solvents to the organic solvent system. Also, there is an advantage of selecting these systems over the conventional formulation approach is that based on our desirable profile, we can tune in the ratio of cation and anion as well as the type of cation and anion. Although there are many advantages associated with these systems, there is one limitation, that is a precipitation of the drug. So, to tackle all these limitations, we are integrating an ionic liquid-based formulation with a supersaturated SNITS approach. As the name suggests, Supersaturated nets are able to maintain the drug in a metastable supersaturated state for prolonged period of time. Thereby, it can synergistically enhance the solubility and maintain the drug in a solubilized state, together tackling the issue of crystallinity and stability. So, basically, this composition is made up of an acid-containing ionic liquid which serve as an oil phase, which is then combined with the optimal surfactant, co-surfactant system and a precipitation inhibitors, together forming a supersaturated net-based systems. So the first objective is to synthesize an ionic liquid. So herein, two different ionic liquids were synthesized wherein the cation was kept constant. So choline was combined with the geranic acid and choline combined with oleic acid, result in two ionic liquid, that is choline geranic and choline oleate. The synthesized ionic liquid was then characterized using FTIR and NMR, where P corresponding to carboxylate ion disappeared and a P corresponding to hydroxyl ion was disappeared. There is a shift in the proton peaks, suggesting ionization has taken place. Once this characterization and synthesis part is done, next is a solubility assessment, wherein solubility study was done by changing the ratio of cation and anion as well as the type of cation and anion system, where it can be seen that the highest solubility was in the cage-based system, which is composed of a ratio of 1:4 of choline and geranic. This solubility was nearly 29,000-fold higher as compared to the water and nearly 2.2-fold higher as compared to synthetic oil-based systems. Moreover, the solubility study was done in surfactant and co-surfactant, since we are making a SNEDS-based system, an emulsion-based system. And upon the screening, the chemophore RH40 and PEG 400 was selected as optimal. Once the component of our formulation is decided, next is optimization was done using iOptimal design, wherein the desirability plot was generated and the predictable and experimental values were compared, whereas an insignificant difference was observed, suggesting the model that was used is significant and it is accurate. Then the morphology of formulation was studied using TEM, wherein the droplet size was spherical. Then, since we are dealing with the emulsion type of system, the stability assessment was done in simulated fluid as well as in the thermodynamic stability evaluation. In both the cases, the insignificant difference was observed, suggesting the formulation is robust and stable. Since we are dealing with a supersaturated nets, one of the major component is a precipitation inhibitor. So the screening and selection of precipitation inhibitor was done using high throughput screening, where it can be seen that in case of different polymers or the precipitation inhibitors, Soluplus and HPMC6 were able to maintain the drug for prolonged period of time. Then the individual optimization was done, where it was observed that over a period of time there is a drastic reduction in the concentration was observed. So instead of going for a single precipitation inhibitor, we went for a combination of two, where it can be observed at 3:2 ratio of HPMC6 and Soluplus. The drug concentration was maintained for prolonged period of time, and thus it was considered as optimal. Next up, the precipitation that occurs in this stage was then analyzed using SEM, where it can be seen that in case of free drug, the definite crystal structure was present, whereas in case of both the formulation approaches, the crystal structure has disappeared, suggesting the drug is either present in molecularly dispersed form throughout the formulation, or maybe it is in the amorphous form. This observation further validated to the PXRD study, where it can be seen that the halo pattern was observed. Then the in vitro characterization, that is simultaneous lipopermeation study, was done using this methodology. So the lipolysis is nothing but the lipid digestion that occurs in the in vivo condition. So in order to mimic that, the in vitro lipolysis study is done, and then the simultaneous permeation study is done. So in this case, it was observed that the highest drug concentration as well as the percent drug recovery was higher in case of our optimized supersaturated net-based system as compared to the conventional oil-based nets as well as the free NIC. Then the apparent permeability evaluation was done, where it can be seen that the two-fold higher permeation was observed in formulation treated group as compared to the conventional formulation or the free drug. Then ex vivo permeation study was done. It was observed that nearly 1.8-fold higher permeation as compared to the free drug. In vitro release study using nanodisk, where it can be seen that the highest release was in the passive condition, which is highly desirable. In situ fusion study was done, wherein effective permeability and absorption constant study was done. Nearly 1.5 to 2-fold higher effective permeability and absorption constant was observed. Once the ex vivo, in situ, and in vitro characterization done, next up is a cell culture based assessment, where it can be seen that the formulation treated group showed highest decrease in the cytotoxicity, highest decrease in the person's cell viability. That means the formulation is cytotoxic enough. Moreover, the IC50 value was significantly reduced in formulation treated group. Next up, quantitative qualitative uptake study was performed, where it can be seen that nearly 1.6 to 1.9 fold increase in the uptake was observed. How it is affecting its apoptosis potential was also done. So in this case, nearly 2 to 4-fold higher apoptosis potential was observed in formulation treated group. The possible reason could be increase in the solubility, permeability, increase in uptake, so the higher concentration of drug reaches to the site and thus increase apoptosis. Viability enhancement parameters, so the pharmacokinetic study was done, where it can be observed that the AUC was improved by 9.88-fold and nearly 1.5-fold as compared to the free drug and the conventional SNET-based formulation. Then in vivo anti-cancer efficacy study was done in syngeneic model, where it can be seen that the significant decrease in the tumor volume, tumor burden, tumor size, and tumor weight was observed in formulation treated group. Moreover, the formulation treated group showed nearly 80% reduction in the tumor growth inhibition, suggesting that the formulation is effective enough. Then in vitro hemolytic toxicity study was done, where it can be observed that the formulation is hemocompatible. Next up, the systemic biomarker level, kidney and liver biomarker level were determined. In this case, it was found that the insignificant increase in both the biomarker level was observed. So the formulation is able to improve the solubility, permeability, oral viability, and it is effective and safer. So the key highlight from this proposed idea and proposed study is that there is remarkable solubility enhancement via ionic liquid based SNET-based system. So the ionic liquid, the NIC showed nearly 29,000-fold higher solubility and 2.2-fold higher solubility as compared to the synthetic oil. The permeation has been improved, the AUC has been improved. This can be served as a prototype formulation approach for the BCS class 2 and class 4, since majority of the drugs that are present in the market belongs to this class. We can tackle the issue of permeability of BCS class 4 with this type of formulation. The present approach that we have used is feasible, it is highly scalable, there is a clinical translation potential, also the IP opportunities and innovation. Further, the future direction, I would like to highlight that we already have a technology transfer of SNET-based formulation to Wow Life Sciences. Moreover, we have an Indian patent of supersaturated SNET-based methodology, as well as for the cage-based formulation as well. So overall, this substantiates that the present approach is clinically highly relevant, and also it gives the opportunity of commercialization. With this, I would like to thank everybody. Thank you so much for paying attention.
I now invite our judges to ask their questions to the presenter.
Since you are using ionic liquids for delivery purpose,
Yes.
so there's a question of regulatory and safety with ionic liquids. How do you justify the long-term oral safety, and because it's a question of safety, then next is the translational perspective, because we are giving it long-term. So how do you see the safety and regulatory approvals from this perspective for ionic liquids? This is one question. Second question, it is connected since it's ionic liquid. It will also impact the permeability of the GI tract, right?
Yes.
So how do you see those questions?
Okay. Thank you so much for this question, sir. So the first question, that is regarding the regulatory concerns and toxicity of this ionic liquid. So we are using a cage-based system, that is a choline, which is an amino acid, and a geranic acid. These are GRAS listed, and they are already present in the market. The cage bio, there is ionic liquid based Samir Mitra Gotri sir that works specifically on cage based system. So already the toxicity profile, the safety evaluation of cage, particularly cage based systems have been done already in the market. Also to substantiate at our lab scale, we have done the toxicity profiling of our formulation. Also the in vitro studies have been done. The data has not been presented, but the formulation of ionic liquid, the cage-based ionic liquid was found to be non-cytotoxic, and then it has been integrated with the supersaturated states. Second question, so can you please repeat that again?
These liquids will also impact the intestinal permeability.
Yes.
So you are
Yes.
looking from a drug perspective, but I'm looking from other than drug also. It will impact.
So in our case, we have to improve the solubility and the permeability. So we are taking that advantages of cage-based system because from the formulation perspective, we want to improve that permeability so that the higher drug concentration reaches to the site, and thus the viability enhancement can occur. So it is beneficial in our case, and also we can study how it is impacting the toxicity profiling in CACO as well. The study have not been performed, but it can substantiate our hypothesis in a better way. So the studies are
Ionic
pending.
liquid, what is the mechanism by which it improves the bioavailability of niclosamide?
So niclosamide belongs to BCS class 2, and it does not have a permeability problem. But the major issue that it deals with is the solubility. Although we are improving the solubility with the conventional formulation, that is oil-based system that are usually used in market, but there is again a problem of precipitation. In vivo condition, in gastric dynamic fluid conditions, the drug often leads to precipitation. This is what we have observed during the experimentation. So in order to tackle that, we are improving the permeation. We want to improve the permeation and solubility, and thereby it is improving directly the viability, because the viability of our drug is mainly limited because of the solubility issue, as it is BCS class 2. So that's why we are integrating ionic liquid for solubility enhancement, and to tackle the precipitation issue, we are integrating it with a supersaturated snake-based system. So they are able to maintain the drug in a metastable supersaturated form for prolonged period. So it will directly have an impact on absorption and thus the viability enhancement. Thank you, sir.
Thank you.
Thank you, sir.
Yes, you have specifically chosen niclosamide. Okay.
Yes, sir.
So, like niclosamide, apart from having your cytotoxicity and anti-tumor effect, it is also an anti-helminthic, right?
Yes, yes, sir.
So why have you specifically chosen? And like, I think you are more focused on the anti-tumor side, right?
Yes, sir. So we
So
are
what?
okay. Yeah, sorry, sir. Continue, please.
You are more focused on the anti-tumor side, going through your experiments. So why have you specifically chosen this particular drug? Why not other drug?
So we have evaluated its efficacy in anti-tumor model because we have this model developed in our lab. However, we are proposing this as a prototype formulation for BCS class 2 and class 4 drug. So we are just proving that how, if we are improving the viability, whether it has the efficacy improvement as well or not. However, the main purpose of this study is to oral viability enhancement. We have chosen niclosamide as a model drug mainly because it has a polymerization issue as well, and majority of the marketed formulation that are present are given in a tablet form, which has 500 mg dose, and also there is a over the period the solubility, there is a changes in the solubility because of its polymorphism. For that reason, we have selected this drug, and to prove whether we are increasing the viability, it has a direct effect on efficacy or not. And since we have a developed model of anti-cancer, that's why we have evaluated the anti-cancer perspective of it. But however, the main aim was to oral viability enhancement formulation, a prototype formulation, and that's why we have selected niclosamide as a model drug. However, this prototype formulation can be used for other BCS class 2 and class 4 drug as well. Since majority of the marketed formulation, the drugs that are present, they have this major tackle issue of solubility and permeability. So we are just proposing the formulation And to extrapolate our study and hypothesis more, we are evaluating the efficacy in anti-cancer. If we have antifungal model as well, we could have evaluated that as well. But for now, we have just focused on anti-cancer efficacy improvement.
Yeah, but my concern is towards your permeability and this one solubility is fine, but my concern is towards your GI. I mean, whether it it will drastically affect the gastrointestinal tract, as Sir Anil has also told. Okay, so you have to look into this aspect also.
Sure, sir. We can do that. Okay.
Okay.
Thank you, sir. Thank you so much.
In addition, in extension of this question, like even hemolytic toxicity has to be considered for the ionic liquids and on the blood cells. What is the effect?
Ma'am, we have not evaluated individually the hemolytic toxicity of our ionic liquid, but the ionic liquid integrated system for the formulation, we have evaluated the hemolytic toxicity. In that case, it is less than 5%. So according to the OECD guideline, it is permissible limit. It is more than less than 2%, in fact, and permissible limit should be less than 5%. Also, we have also evaluated the morphology of our RBCs. So in that case as well, the morphology was similar to the PBS. So we have not evaluated of cage individually, but the formulation we have evaluated based on systemic biomarker level and hemolytic toxicity. Also, we are carrying out the histopathology studies as well.
Okay, thank you.
Thank you. Thank you.
Thank you. Now, I would like to invite code number PhD 022. Please be ready. We'll be playing your video shortly. Please switch on your cameras.
Presenter, please switch on your camera. Code 22, please switch on your camera, please. Okay.
Good afternoon, respected examiners and all participants. So I would like to begin with a scientific thought by Wilson and Menard. The therapeutic strength of the phytoconstituent is not determined by its concentration, but by its capacity to reach the target tissue in an active form. The perfectly capture core challenge in phytopharmacology. Our compounds are not weak, our delivery is. Our research addressed the gap by developing bosomes, a phytophospholipid complex developed to enhance hepatic delivery and unlock multi-mechanistic hepatoprotection. Let's understand the global problem that makes this study essential. Alcoholic liver disease is not just a biochemical disorder, it's a rapidly accelerating global crisis contributing to nearly 3.3 million deaths each year. Although herbal phytophenols possesses remarkable hepatoprotection and their therapeutic benefit fails to reach clinical success because of poor solubility, low permeability, and rapid metabolism. So we asked a simple question: can we convert this bioactive molecule into a suitable and highly deliverable form that reaches the liver more effectively and work more powerfully? And yes, the question leads the birth of bosomes. Now that the problem is clear, let's see how we design scientific solution. So instead of random trial and error formulation, we adopted central composite design based quality by design approach. We systematically identified phospholipid content, temperature, and reaction time as a critical material attributes, while particle size, zeta potential, entrapment efficiency, and PDI was selected as a critical quality attributes. With 20 experimental runs, the model demonstrated excellent statistical robustness, excellent R-square value, non-significant lack of fit, and strong predictive power, giving us complete confidence in optimizing this formulation. Further, the overlay plots make the story very clear. This yellow region indicates or denotes the multi-dimensional optimization with all critical quality attributes meets the desirable criteria simultaneously at the precise point of phospholipid to fraction ratio 1:3 weight by weight and the temperature 50 degrees Celsius. We achieve optimized bore zone delivering 194 nanometer particle size, a PDI below 0.3, zeta potential close to minus 30 milliwatt, while entrapment efficiency more than 94%. In simple words, the formulation achieves solubility, uniformability, and maximum durability all at once. Every characterization tool for the confirmed successful Borzone formulation, like HRTM, visualize uniform and spherical nanoscale particle size, while in vitro release profile showed sustained and controlled release pattern supported prolonged availability of active fraction at the target site. FTIR clearly demonstrated new hydrogen bonding between phytoconstituent and phospholipid, while PXRD showed a complex disappearance of crystalline peak, providing the shift to an amorphous state responsible for enhanced solubility. Importantly, 1H NMR exhibited a significant field shape of the triethylamine proton of phosphatidylcholine, confirming the shielding due to close association with the phytoconstituent. Together, these results confirm that fraction is not a simply entrapment of lipid base, but chemically structured with the phytophospholipid and formed a complex. Now come to the exciting part, that is efficacy. So in ethanol cetyl Hep2 cells, Borzone shows remarkably powerful hepatoprotection even compared to the standard drug that is silymarin. They restore cell viability and restored nitric oxide while enhance the detoxifying enzyme ADH and LDH. Simultaneously, most importantly, Borzone shows significant reduction in lipid accumulation in the Hep2 cell while also shows strong antioxidant effect. This data indicates the coordinated modulation of oxidative stress, inflammatory marker, and metabolic pathway at cellular level. For any formulation have clinical relevance, it must be safe. So after 28 days of repeated oral dosing, there were no alteration in the biochemical, hepatological, and histopathological parameters. This confirmed that bozones are the biocompatible, non-toxic, and suitable for chronic therapeutic use. Further, in alcohol-induced ALD model, the therapeutic advantage become more striking. Borzomes dramatically reduce liver indices, spleen indices, and the suppress NF-kappa B and pro-inflammatory cytokinin level. At the same time, they restored plasma liver functional markers and hepatic triglyceride and significantly evaluated endogenous antioxidant defense by increasing the level of GSH, catalase, GP and SOD while reducing the level of MDH. So in simple terms, Borzomes prevented inflammation, oxidative stress, and lipid accumulation simultaneously by supporting the previous cellular level data. To mechanistically substantiate the therapeutic effect, we analyze hepatic gene expression in same ALD model. And Borzone significantly upregulated the AMPK, NRF2, SRT1, and PPRY gamma level, indicating activation of energy regulation and antioxidant defense pathway. At the same time, Borzone markedly downregulated the level of TNF-alpha, CYP2E1, TLRF4, and SREBP1, which reflecting suppression of the inflammation, ethanol-induced oxidative stress, and lipogenesis as well. This dual mechanism increased the protective gene and decreased the pathological gene, demonstrated a coordinated molecular reversal of the ALD. Overall, all the gene signatures confirm the Bohr polysome acts the transcriptomic level to restore hepatic hemostasis rather than providing systematic relief. So let's move into our key findings. So to conclude, Bohr polysome represents scientifically rational, mechanistically proven, and clinically transformable approach for the management of alcoholic liver disease. With this research, we demonstrate the phospholipid complexation with supported quality by design approach can convert promising herbal fraction into potent, safe, and transformable therapeutic system. So, home taking message is that Bohr polysomes are not just a formulation, they are step forward, reflecting the future for the hepatoprotective therapy. Thank you.
Thank you.
Since you have made a formulation
I
from
now
that
invite our
herbal
judge.
extract, right?
And now invite our judges to ask their questions to the presenter. Thank you.
So you have made a formulation of herbal extract, right?
Yes,
Are you
sir.
getting? Yeah.
Yes,
So
yes, sir.
a liver disease will have different stages clinically.
Yes, sir.
Right. So where you see your formulation will work, whether in steatosis, hepatitis, fibrosis, or cirrhosis, at what level of clinical relevance?
So actually in first stage, that is steatosis, where lipid accumulation and enhanced oxidative stress as well with inflammation, like first stage on steatosis.
But most of the clinical cases, people will even go underdiagnose the stage 1 because the clinical impact biochemically results in a very later stage. Because liver
Yes,
is a
sir.
very important organ, right? And it regenerates also its cells. So how do you see, like, how does this is one question was there. Second question is, how does ongoing like alcohol consumption in patients affect the bile secretion and enterohepatic circulation that will affect your this borosome formulation?
Thank you for question, sir. Actually, sir, I have worked on the first stage of alcoholic liver disease, that is steatosis, where the lipid accumulation, oxidative stress, and inflammation will occur to liver. And in early stages, people will not find these symptoms, but after cirrhosis, they will find that liver. After like cirrhosis, they will find these symptoms, and these bosomes will act very longly. Like we have did bioavailability study as well. And second thing, we have used Lieberman Decarly diet model in which generates the shows the total human-like tendency in the liver.
And second part, like those people who are taking alcohol consumption, that will impact the enterohepatic circulation as well as with the bile secretion, right? So
Yes, sir.
that in those contexts where you see your formulation, that will also
Sir,
change the course of the delivery, bioavailability, and all these things.
yes, sir. Sir, we have checked liver markers like ALT, AST, and all other liver markers, which are due to imbalance in bile secretion, they also alter. So this prepared Bohr polysome shows significant increase in these biomarkers. So yes, definitely they will reduce bile level as well,
Fine.
which
Thank you.
significantly increase in alcoholic liver disease.
Thank you. Over to you, Nanavati sir.
So you work on Borazome, and I
Yes.
think Borazome is a commercially available formulation, right?
No, sir. Actually, I have given name because the phytosomes, it is a copyrighted name. So we can't see
Okay.
phytosomes. That's why I have named this my formulation as a borzomes.
Okay, fine. So it's
Yes.
particularly a multi herbal extract.
Sir, not a multi-herbal. Actually, I have prepared one enriched fraction from
Okay.
plant Borrelia diffusa. Yes,
Okay.
sir. Rotenoid enriched fraction.
Okay. So you
A
have worked
particular
on
type
this.
of compound. Yes, sir.
Okay. So you have worked on the antioxidant part, right? So
Yes,
what
sir.
are the protocols which we have used? Number one. Number two, for assessment of this oxidative stress,
Yes, sir.
what has been done regarding that?
Sir, I have for oxidative stress, I have seen first the oxidative stress parameters like SOD and other NRF2 in the serum level as well, and in the gene level as well. Like they are acting on gene mechanistically, they are reducing NRF2 level and increasing antioxidant mechanism in body. Like they are enhancing SOD level, then MDA and reducing MDA level and other things,
Then yeah.
which I have
And
evaluated in plasma and mechanistically.
okay. And what about the antioxidants? What are the methods which we have used?
Sir, first I have checked on cell
In
level
vitro.
using
Yeah.
DCFDA assay, and
Okay.
for serum I have used these parameters.
And what was the control?
The control I have taken Silimarin, which is already in market drug.
Okay, so as you compare the IC50 of Silimarin and your formulation, the IC50 of your formulation is lower.
Yes, sir.
Okay, good. Thank you.
Thank you, sir.
Thank you, judges, for your insightful questions. Now, we request the judges to share a few words about their experience on today's event. Thank you.
Anil sir, if you could give the conclusive remarks.
Yeah. Okay. Thank you, first of all, for giving this opportunity. Again, last year also, I think we both of us were evaluating the candidates. And this time also we had a good experience. Actually, we also learned from those presentations because we may not be expert in each of this, but we also try to understand those and try to. So the questions are more from basically learning perspective rather than just to squeeze them or asking them. So overall, it was a good experience. We had a good presentations, and once again, thank you for giving this opportunity. Can you just confirm once that the 020 is not there today? The 020? Because the list what we received, there was one more presentation.
The candidate, yeah, the presenter must have not been present in the
You can mark him absent, him or her, whoever, right? Okay, thank you once again.
Excuse me, sir, but participant 020 is disqualified.
Is it disqualified? So it's not there, right?
Yes, sir.
Okay. Thank you. Thank you. That's all from my side.
Yeah, Dhanraj sir.
Good afternoon once again. I'm rather very thankful to Bhanuben, Nanavati College of Pharmacy. It's been second time here. Both of us, I mean, Anil sir and me, we were there last time also on the 5th Congress. And today also on this 6th Congress, we are here for evaluation of the presentations. The presentations were met quite well, but somewhere not so good, but they need improvement. And students should be very prompt, whether they are presenting or not. And, some of the students were missing during the questioning round. Of course, we're not going to give marks for that. But overall experience was good. And we also got many things which we learned from all those presentations because we are not expert of all the fields, right? As Anil sir has also told. So in future also, we do hope that such kind of Congress will happen and at a very, improved and larger scale. And, the interest of the student should be there in such kind of organization of this Congress. It is not only the teachers' initiative that should be looked up. Students also have to participate. They also have to, prove themselves that they are capable of organizing, they are capable of presenting, not only research, research to hota rahega, it'll keep on moving. But, interaction is very much required and exposure is required. Thank you very much. Thank you for the wonderful experience.
Yes, thank you, Dr. Dhanraj. That was a good suggestion. And yes, last year also you were with us, and we would be continuing every year Student Research Congress. We see every year more and more universities are taking part, and we see better, the quality is improving, better institutes are taking part. The credit goes to all our judges who have been giving their wonderful insights and giving a good judgment and identifying the right winner. Even the last SRC, fifth SRC, it was like truly national, where we had winners from all four corners of India. And this time also, let's see how the winners go. But the judges definitely play a very important role. So we sincerely thank you and Dr. Anil for supporting us for this. Thank you very much.
Thank you. Thank you, ma'am.
Yeah, over to you, team. Vasanti, madam.
Should we have a group photo for this particular event? Or it's already taken?
Ma'am, we can take the group photo.
Okay, you've taken, right?
I request all the participants to please switch on their cameras. We would be taking a selfie right now.
All right, perfect. Over to you, Afrin madam, for the final words and then the valedictory when we move.
Thank you, ma'am.
On behalf of the entire organizing team of 6th SRC, it is our privilege to deliver the vote of thanks. First and foremost, a heartfelt gratitude to our esteemed judges for their time, expertise, and thoughtful evaluations. Your guidance and feedback have made this session deeply enriching. A special appreciation goes out to all our participants. Your passion, dedication, and innovative ideas have made this event truly inspiring. You are the heart of this competition, and we celebrate your hard work and achievements. Finally, sincere thanks to our audience, faculty members, organizers, technical team, and everyone present. Your constant support and encouragement have contributed immensely to the success of this event. Thank you all, and wishing each one of you continued excellence, creativity, and growth in your research journeys.
We now look forward to the awards and valedictory session, where we will recognize and celebrate the exceptional contributions and achievements of our participants. The session is beginning shortly, and the meeting link has been shared in the schedule and the chat box. Dear judges, as we have other presentations still running, we are starting the valedictory session at 2 p.m. We shall be calling you all personally to come form, requesting you all to please be present for the valedictory session. Thank you.
Vasanti madam, do you want to have a separate meeting, three of you? So I will ask Afrin madam to create one meeting room where three of you can discuss. So you can manage. Vasanti madam will take the lead on that.
Yes, ma'am. Thank you.
All right. Okay. Thank you very much, all the judges, Dr. Anil and Dr. Dhanraj. We look forward to see you in a valedictory session, because we want you